Biotechnology is the science that lets humans redesign life itself. Let's start biotechnology principle and process. What is biotechnology? According to the European Federation of Biotechnology, or EFB, biotechnology is defined as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Biotechnology has two main components. First, genetic engineering. These are techniques used to alter the chemistry of DNA or RNA and introduce it into a host. This results in a change in phenotype. Second, bioprocess engineering. It involves the maintenance of a sterile, meaning microbial contamination-free, ambience. It is used in the production of antibiotics, vaccines, and enzymes. Next topic: the birth of modern biotechnology. In 1972, two scientists, Stanley Cohen and Herbert Boyer, gave birth to modern biotechnology. They worked with a bacterium called Salmonella typhimurium. Inside it, there was a native plasmid carrying an antibiotic resistance gene. Now, what is a plasmid? A plasmid is a small, circular DNA that can replicate on its own. Think of it like a delivery truck carrying genetic information. First step, cut. Scissors. They used special enzymes to cut the plasmid DNA. Second step, paste or link. They inserted the desired gene into the plasmid. This created a new DNA molecule. Third step, transfer. The modified plasmid was transferred into another bacterium, E. coli. Now, this bacterium carried recombinant DNA, rDNA. And that was the beginning of genetic engineering in modern biotechnology. Next topic is the enzymes, the scissors. Restriction enzymes belong to a class of enzymes called nucleases. There are two types of nucleases. First, exonucleases. Exonucleases remove nucleotides from the ends of DNA. Second, endonucleases. Endonucleases make cuts at specific positions within the DNA molecule. Did you know? The first restriction endonuclease discovered was Hind2. It cuts DNA by recognizing a specific sequence of 6 base pairs. Next topic is how restriction enzymes cut. Have you ever wondered how restriction enzymes cut DNA? Let's understand the concept of palindromic nucleotide sequences. A palindromic sequence reads the same forward and backward, just like the word Malayalam. For example, look at this DNA sequence. 5 feet, got TC, 3 feet. 3 feet, CTTAAG, 5 feet. Notice something? When read in the 5 feet to 3 feet direction on both strands, it reads the same. That's why it is called a palindromic sequence. Now, restriction enzyme EcoRI recognizes this specific sequence, got TC. It cuts between G and A on both strands. After cutting, it forms sticky ends. These sticky ends have overhanging bases that can form hydrogen bonds with their complementary sequences. This helps DNA ligase to join DNA fragments together easily. Now, let's understand the naming of EcoRI. E stands for Escherichia, the genus. Co stands for coli, the species. R stands for the strain, RY13. And I represents the order of isolation. So, restriction enzymes recognize palindromic sequences, cut them, and create sticky ends, which are very useful in genetic engineering. Next topic: cloning vectors, the vehicle. DNA. What are cloning vectors? Cloning vectors are DNA molecules that carry foreign DNA into a host cell and replicate there. They contain special sequences that allow them to be recognized by the host cell machinery for replication. Repeat button: common vectors. Plasmid. Bacteriophage pushpin must have features of a vector. Keycap 1 origin of replication, ORI. It starts replication and controls the copy number. Keycap 2 selectable marker. It helps identify transformants. Example: ampicillin resistance gene. Keycap 3 cloning sites. These are recognition sites for restriction enzymes. And remember, they must be unique. Bullseye closing line. So, a cloning vector acts like a vehicle, carrying the desired gene safely into the host cell and multiplying. Next topic: the MVP vector, PBR 322. Here, P stands for plasmid, BR stands for Bolivar and Rodriguez, and 322 is the identification number. Studio microphone main content: PBR 322 is a circular plasmid vector. It contains two important antibiotic resistance genes, AMPR, for ampicillin resistance, TETR, for tetracycline resistance. These help in the selection of transformed bacteria. It also contains ORI, the origin of replication, which helps the plasmid replicate inside bacteria. ROP gene, which regulates plasmid copy number. Studio microphone restriction sites present. PBR 322 has several restriction enzyme sites such as EcoRI, CLAI, Hind III, BAMI, SALI, PSTI, and PVUI. These restriction sites help in inserting foreign DNA into the plasmid. Studio microphone conclusion. Because of its selectable markers, origin of replication, and multiple restriction sites, PBR 322 is considered the MVP vector in genetic engineering. Next topic. How do we know if our recombinant DNA experiment actually worked? How do we tell recombinants from non-recombinants? There are two main methods: the hard way and the easy way. Bullseye 1. Antibiotic plating, the hard way. First, we insert the foreign DNA at the BAMI site. This insertion disrupts the TETR gene. Now what happens? Right finger recombinant bacteria will grow on ampicillin. Right finger, but they will die on tetracycline. So, on the ampicillin plate, colonies grow. On the tetracycline plate, recombinants die. This method works, but it requires replica plating and careful comparison. That's why it's called the hard way. Bullseye 2. Color selection, the easy way. Here, the gene is inserted within the beta-galactosidase gene. This insertion inactivates the enzyme. Now observe the colonies on a chromogenic substrate plate. Right finger if there is no insert. Beta-galactosidase is active, right-pointing arrow colonies turn blue. These are non-recombinants. Right finger if the insert is present. Beta-galactosidase is inactivated, right-pointing arrow colonies remain white. These are recombinants. So, simply select the white colonies. Easy, clear, fast. Bullseye concept summary. Insertional inactivation helps us identify recombinant plasmids. If a gene is disrupted due to foreign DNA insertion, its function is lost, and that loss helps us detect recombinants. Next topic: vectors for plants and animals, disarming the pathogen. In biotechnology, we use vectors to transfer genes into plants and animals. But how do we do that? We actually disarm pathogens and use them as tools. Seedling for plants. The bacterium Agrobacterium tumefaciens naturally infects plants. It delivers a special DNA segment called tDNA, also known as tumor DNA. Normally, this tDNA causes tumors in plants. But scientists remove the tumor-causing gene and replace it with our gene of interest. This modified bacterium then transfers the desired gene into the plant cell. The result? A transgenic plant. PA for animals. In animals, we use a retrovirus as a vector. Retroviruses naturally transform normal cells into cancer cells. But scientists disarm the virus by removing harmful genes. Then, we insert the desired gene into the virus. This modified virus delivers the gene safely into animal cells. Sparkles. So in both plants and animals, we take harmful pathogens, disarm them, and turn them into powerful tools for genetic engineering. Next topic. Tool number 3. Competent hosts DNA problem. DNA is hydrophilic, membrane is hydrophobic. So DNA can't pass through the membrane. To solve this, we use different methods to make host cells competent, meaning ready to take up recombinant DNA. Small diamond method 1. Chemical heat shock. First, treat cells with calcium ions, Ca². Keep them on ice. Then give a heat shock at 42 degrees Celsius. And again place them on ice. This sudden temperature change helps DNA enter the cell. Small diamond method 2. Microinjection. In this method, DNA is directly injected into the nucleus using a fine needle, mainly used for animal cells. Small diamond method 3. Biolistics, gene gun. Here, gold or tungsten particles are coated with DNA. These particles are shot into the cells using a gene gun, mostly used for plant cells. Small diamond method 4. Disarmed pathogens. Modified pathogens are used as carriers to transfer recombinant DNA into host cells. Sparkles. All these methods help host cells take up recombinant DNA. This process is called transformation.